Am I clear to all of you? Have you understood the salient features of this histidine operon?
Yes.
So tell me, let me know some of the, let me know the some important points which which I have described, described now. Point out some of the features which I have just pointed out. Any
So
one of
it
you?
has touch structural gene, and 2 gene
Yes,
encodes the same enzyme.
yes,
It has multiple regulator, and it comprises of 5 regulator gene, and they work together. There are 5 regulator gene RUSTW, and
yes,
location of promoter is not defined.
yes,
Uh, and attenuation occurs,
yes. It is
and
connected to
repressor
attention. Yes,
and it also works as aporepressor. But, it has aporepressor, and histidine also works as co-repressor, acts as
yes.
co-repressor.
What is your name?
Tushika Sinha.
Very good, Tusika. You have explained the, you have pointed out the important features of the histidine operon, which I have discussed today in this lecture, and I think that you are following this lecture in a very good manner, and I think that all of you should participate or should interact when when I suggest you, when I tell you, when I ask you to interact with me. See, I am very happy with you because you are responding to me and you are following the lectures given, lectures delivered. So just again, and I expect the same thing from all of you who have joined this lecture, because you know that this all one of the very important component of online teaching is the interaction of the student with the teacher. So yeah, so I expect the same uh, the same thing from all of you. So now look at the whiteboard and try to understand the location of the different genes on the different genes in case of histidine operon. So just I would like to show you the whiteboard on which the uh, histidine operands has been drawn. Look at the whiteboard. Let me know whether this whiteboard is visible to all of you. Tusika Sinha,
Yes, sir.
देखो, this is I have shown here. This is the bacterial genome. This circular line represent the bacterial DNA, the genomic DNA, and on this genomic DNA, I have shown this regulator gene in the reading R R U S T R U S T and W. These 5 alphabets indicate the 5 regulatory genes located on the genome of the Salmonella typhi murium. And RUST, these 5 genes are in sequence. And just I have told you that the location of the promoter is not very clear, is not very well defined. And it is still controversial. There are two school of thoughts about the location of this promoter region. According to one school of thoughts, the promoter is located in between the regulator genes. Just look here, this promoter has been shown in between the T and W. So these are the 5 regulatory genes R, U, S, T, and W, and the promoter is being shown uh, among in between the T and W. So this, according to one school of thought, the promoter may be located in the among the regulatory genes, and according to the another school of thoughts, this promoter is present between the operator and these regulatory genes. Okay, so there I have shown both the conditions, and either of these two conditions may be present on this genome. And look here, there are 10 structural genes, and the 10 structural genes have been shown in the numerals 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. These numerals indicate the 10 structural genes located in the histidine operon. And the same thing I have shown here. Look, this straight line shows the DNA on which the RUST. These are the four regulatory genes are in sequence, and there is W, and in between the T and W, the promoter gene has been shown. It may be the condition, or if these five regulatory genes are in continuation, the promoter may be present here in between the operator and regulator genes. And there are 10 structural genes, shown here, one in the numerals starting from 1 to 10. And look at these five, the very important features which I have just discussed. This histidine operon was reported in Salmonella typhimurium. This is an example of repressible operon. This is negatively controlled. You know that lac operon is both positively and negatively controlled. Tryptophan operon is negatively controlled, and this histidine operon is also negatively controlled. Presence of the special feature of this operon is that there is presence of multiple regulators consisting of 5 regulatory genes, presence of 10 structural genes, location of promoter is controversial. There exists coordination. There exists coordination among the five regulatory genes which act as a single unit. And if there is any mutation in any of the 5 regulatory gene, the regulator will become non-functional. Okay? The 10 structural genes encode 9 enzymes, which shows that there are 2 structural genes which encode the same enzyme. There is absence of collinearity between the sequence of the genes, sequence of the structural genes, and sequence of biochemical steps. What does it mean? It means that you know that in case of lac operon and tryptophan operon, there are in case as and try to understand. In case of lac operon, the structural gene 1 encode beta galactosidase, the structural gene 2 encode the lac permease, and the structural gene 3rd encode the acetylase. You know that there are three genes, structural, and 3 enzymes, and the 3 steps are catalyzed by one by one. So in case of a tryptophan operon, there are 5 structural genes encoding 5 enzymes, and the first enzyme encode the first step, the second enzyme encode the second enzyme catalyzes the second step, the third enzyme catalyzes the third step, and the fourth and fifth enzymes catalyzes fourth and fifth steps respectively. But there is no such collinear relation. There is no such collinear relation among the sequence of the structural genes and the enzymes encoded by 10 structural genes and the 10 steps required for the biosynthesis of this histidine from its precursor. What does it means? It means that this you can understand that the enzyme encoded by the 10th structural genes catalyzes the first step here. Try again to understand what I am saying here. Is the 10th structural gene encodes an enzyme, and that enzyme catalyzes the first step of the biochemical pathway of the histidine biosynthesis. The first, it is not the case like this कि ऐसा नहीं है इसमें कि पहला स्ट्रक्चरल जीन जो एंजाइम बनाता है वो फर्स्ट स्टेप को कैटलाइज करता है. This is not the case. The case is different, that the enzymes encoded by the 10th structural gene catalyzes the first step of the histidine biosynthesis. Therefore, just I have mentioned here that there is no collinear relation between the sequence of the enzyme, between the sequence of the structural genes and the enzymes, and the steps of the biochemical pathway of histidine biosynthesis, because the 10th structural gene encodes the enzyme, and that enzyme catalyzes the first step of histidine biosynthesis pathway. This is not the case in case of tryptophan operon. In this way, it is different from the tryptophan operon. Okay? The regulator encodes inactive repressor, which is known as the aporepressor, and the histidine acts as a co-repressor. Like these 2 points and these 2 features are similar to that of the tryptophan operon. because in case of tryptophan operant, you know that the regulator encode the inactive repressor, which is known as a co-repressor, and the tryptophan act as a co-repressor. Similarly, in this case also, the repressor encoded by the regulator of this operant is also inactive, and the histidine itself act as a co-repressor. And the very important point I would like to mention here, that these five regulatory genes work together. If there is any, and all these five regulatory genes will act together to encode the repressor protein. The coordinated functioning of these five regulatory genes are required for encoding the repressor protein. And if there is any mutation in any of the five genes, the regulator will become non-functional. So these are the salient features of the histidine operant. In the next lecture, we will be studying the this is the structure. In this lecture, we have studied the salient features and structural organization of this histidine operant. Am I clear to all of you? Am I clear to all of you?
Yes, sir.
This lecture has
सर कोरिलेशन किसको बोलेंगे? कोरिलेशन
आवाज नहीं आ रही।
सर कोरिलेशन किसको बोलेंगे?
हिस्टिडीन वो
नहीं मतलब वो कोरिलेशन क्यों बोला जाता है?